This is part 2 of the walkthrough of the AQA GCSE Combined Science Trilogy 2022 Biology Higher Tier Paper 1. Question 4. Photosynthesis is an important chemical reaction in plants. 4.1. Why is light needed for photosynthesis? Right, so photosynthesis is a chemical reaction. It needs light for it to work. Why? Well, light is a source of energy, so the light needs to be there for energy for the reaction to work. But the other thing that we can remember about photosynthesis is that it is an example of an endothermic reaction, meaning that energy is taken in from the surroundings. So that energy will be light energy. 4.2. What is the equation for photosynthesis? Tick one box. So they've given us it as symbol equations now. So we've got to remind ourselves about our symbols. So the first thing about photosynthesis is that its role is to make food for the plant, which is glucose. And our formula for glucose is C6H12O6. So that first equation can't be correct then, because our formula for glucose is on the reactant side, so not right. Looking at our second equation, glucose formula is not there. So our glucose formula is there for our third and fourth one, and it's on the product side, so we know we're in the right lines. But what's the next thing that we have to think about for photosynthesis? A plant needs to also take in carbon dioxide for the process to work. So if we look at our fourth equation, carbon dioxide is a product, so that can't be right. So the third box should be our answer. 4.3. A student investigated the effect of different colors of light on the rate of photosynthesis at room temperature. The student used pondweed in water. A piece of pondweed was placed in red light, then in blue light, and then in green light. Each color of light was the same intensity. Describe how the student should make accurate measurements to obtain valid results for the rate of photosynthesis. 4 marks. So a lot of information is given to us here. Let's break it down step by step. First thing that we need to do, ultimately the question is asking us to refer to these terms, accurate and valid. So the definition of accurate means close to the true value. The definition of valid really is related to reliable. So in essence, the question is asking us, how are we going to do something in this experiment to ensure that the results that we get are reliable and close to the true value? Well, let's look at that first sentence then again. And thinking back to our previous question, which is to do with our amylase, it's talking about, again, the effect of something. So we're investigating the effect of different colors of light. So our independent variable will be the different colors of light. That's what we're going to be changing. But what are we going to be measuring? We're going to be measuring the rate of photosynthesis. But how do we do that in the experiment? What pieces of equipment will we use to measure this rate of photosynthesis? We don't have a specific piece of equipment to measure the rate, but we can get there with some pieces of equipment. So for photosynthesis, there are two common ways in which we can do this. The first thing that we could do is we could count the number of bubbles of oxygen that are produced. And the second way that we could do it is by measuring the volume of oxygen that is produced. So we have these two methods that we could talk about, but again, We want to obtain valid results and make accurate measurements. So which one of these two is more likely to do that with the less errors involved? Is most likely going to be the volume of oxygen produced because of the pieces of equipment that we could use. So we know the method that we're going to go down now. We've got to think further about how we can make some accurate measurements. So in essence, we've spoken about our independent variable, our dependent variable. What about the control variables? Well, thinking about photosynthesis and rate of photosynthesis, there are three things that can affect the rate of photosynthesis, and these are our limiting factors. Temperature. Well, they said in the question, first sentence, it's been used at room temperature. The light intensity. They said in the fourth sentence, the color of light was the same intensity. So our third factor has not been mentioned, carbon dioxide concentration. So what we can do is we can state how we can keep the carbon dioxide concentration the same. And we can do that by using a specific chemical. And that chemical is called sodium hydrogen carbonate. So the first thing that we can say then is by using the same concentration of sodium hydrogen carbonate, because that will provide a constant concentration or constant amount of carbon dioxide. So we know if we're using the same temperature, same light intensity, and same carbon dioxide concentration, then the only thing that is going to affect our result will be the color of light. Then we can start to talk about the method a little bit. We're going to use a measuring cylinder to measure the volume of oxygen produced. So we've made it clear in our answer that we know that we're trying to measure something accurately by using a piece of equipment that has values, as opposed to counting with our eyes. The next thing that we got to think about then is if we have collected the volume of oxygen produced and measured it, that is not going to give us the rate, because rate is always something divided by time, something over time. So it would mean that we would also need to record time in which this oxygen is produced and the volume is measured. So again, piece of equipment that we're going to use to measure the time will be a stopwatch. So we'll say that we'll use a stopwatch, and then we would measure this volume of oxygen produced in a certain time. So for example, in 2 minutes or 3 minutes. So let's say, for example, that we collect our oxygen, measure it in the measuring cylinder, and we find out that we've got 30 centimeter cubed of oxygen produced, and we have taken that measurement over a period of 3 minutes. What we would then do is just take that 30, divide it by 3, and then we have our rate, 10 centimeter cubed per minute. So now we've got to think about what are we going to do when we collect these results. Well, what is one way to improve accuracy in any experiment? The most common thing that we can do is take repeat measurements. So at each color of light, so for example at red light, we would do the experiment at least 3 times. Once we've done that, we've got 3 sets of values, we can calculate a mean. But when we are looking at those values in order to calculate the mean properly, we want to identify the values that are not following the pattern. And those are called anomalies. So any values that are not following the pattern, we can remove them, exclude them from our calculation of the mean, so it doesn't affect the mean too much. A scientist investigated the effect of different wavelengths of light on the rate of photosynthesis. The wavelength of light determines the color of the light. Figure 8 shows the student's results. Figure 9 shows the scientist's results. So let's look at figure 8 first. You can clearly see that we've got a bar chart there. Let's look at figure 9. We can see that we've got a line graph there. 4.4. Why are the results for the two investigations presented differently? 2 marks. So yes, why is one graph a bar chart and one graph a line graph? Well, again, we've got to look at what is being investigated, what's being measured, and that will give us an indication of why the graphs are different. So from the students' results, you can see that they have got 3 different colors of light and the rate of photosynthesis is measured. But the red, blue, and green are separate. They're in different categories. So any time that we use a bar chart is because some of our data is in categories, and we classify that as discrete or discontinuous data. Looking at figure 9 now, again we've got rate of photosynthesis, but our x-axis is the wavelength of light. Now here it's not in different categories because we can have any value between 400 and 500, 600 and 700. So that data is known as continuous data. So we just need to state that the student's results are in categories, so the discrete data. The scientist's results are continuous data. 4.5. Suggest the range in wavelength of green light. Use figure 8 and figure 9. 1 mark. So we've got to give a range, so 2 values for this range of green light. So they said use figure 8 and figure 9, so let's go to figure 8 first. We can see the green light has given us the lowest rate of photosynthesis. So if we want, we can find out what that actual value is and see if that helps us. The value of our rate of photosynthesis for green light is going to be 0.6. So we know that value then. Let's go to our figure 9. Well, 0.6 is going to be very difficult to find on this figure 9, but we know that it's going to have the lowest rate of photosynthesis. So it seems like we don't have to use the value, we just need to use the fact that it's the lowest rate. So we can see there's 2 peaks on our graph here. So those 2 peaks would probably represent the red and the blue light. So this lowest area has got to be our green light. So we've just got to find our lowest value of wavelength and our highest value of wavelength, and then that would be our range. So we can see the point where it starts to go down earliest. If we read that down to our x-axis, that looks like it's going to be about 480 nanometers. And the point where it's just about to start increasing again, we can see that that's just before 650. So we can say probably 645 nanometers. So our lowest value for our range would be 480. Our highest value for our range is going to be 645. And that would be our answer for this question. Question 5. This question is about tumors. 5.1. Describe the similarities and differences between benign tumors and malignant tumors. 4 marks. So reminding ourselves about what tumors is linked with, it's linked with the term cancer. So let's define cancer first, and that is uncontrollable cell division. So when the cells are dividing more than they need to, you then get overgrowth of cells, and that becomes a tumor. What is this caused by? What causes this uncontrollable cell division? Well, it's through mutations, which are changes in DNA. So we need to remind ourselves, how are these two types of tumors, benign and malignant, similar, and how are they different? Well, let's use our information that we've gotten already. Both of them are caused due to mutations in DNA. But then these mutations both result in uncontrollable cell division. So already we've gotten two similarities. So we just need to add some differences now, and we should be able to get our full marks. Now, when people think about cancer and how much of a problem it is, they're really thinking about malignant tumors. Now, the reason why cancer is such a big problem, especially when it's malignant, is because it's able to spread to other parts of the body very quickly. Whereas benign tumors, they stay in one location, which is why it's easier to actually treat. If someone has a benign tumor, they can just go through a surgery where it just gets cut out. Whereas someone has malignant tumors, they're going to have to go through either chemotherapy or radiotherapy. Now, why is it that this benign tumor is not able to spread like the malignant tumor? It's because this layer of cells is surrounded by this layer or a capsule, and that is causing them to be fixed in a certain location. Whereas the malignant tumors, they're not surrounded by anything, so they're able to just move once the cells just grow more. The other thing that we can talk about in terms of the tumors are its difference in speed of growth. The malignant tumors are going to grow faster than the benign tumors. And the last thing that we can add is that malignant tumors, because they're able to spread, they can form secondary tumors, so tumors in another location. And the benign, because it's not able to move, as I said, it's surrounded by this capsule, this layer, it's not able to form secondary tumors. Figure 10 shows some data about skin cancer in males and females for different age groups in the UK. It shows the number of new cases of skin cancer in 1 year, the number of people with skin cancer per 100,000 population in 1 year. So let's look at figure 10 quickly. You can see it's got a lot of information on there, and it's got two y-axes. So that means that we've got a lot of data that we need to analyze carefully. A key is given to us. So the bars are representing new female and new male cases, and the lines represent the number of males and females with skin cancer per 100,000. So what we can do with this key is we can figure out which axis is responsible for which part of the graph. So we know that the dashed line and the solid line is for the y-axis on the right, whereas for our bars, that is going to be applying to the y-axis on the left-hand side. So when it comes to analyzing this, we can do this a little bit faster knowing this information. 5.2. There are no new cases of skin cancer diagnosed in people younger than 15 years of age. Explain why. 2 marks. So, if we go back to our graph now, you can see that our lowest age is 15 to 19, where there's nothing else below that. So why is there nothing else below that? It's already very low at that age group, 15 to 19. Why is there nothing recorded before that? Well, let's think about skin cancer itself. One of the most common causes of skin cancer is through exposure to radiation. specifically ultraviolet or UV radiation. So the more exposed you are, the more likely you are to develop this skin cancer. So someone that is younger, less than 15, is not going to be exposed to much radiation because they haven't been alive for that long. So that's probably something that we can mention. People younger than 15 are less exposed to radiation compared to older people. But we've got to think about a second mark, and it says we've got to explain. So what might be some further information that we could add? Well, again, what is cancer due to? Cancer is due to mutations, changes in DNA. So that means that there's less of this change in the DNA. So there's less damage that occurs to the DNA. So that would mean then that there is less chance of developing cancer. 5.3. Give two conclusions about the number of new cases of skin cancer. Use figure 10, 2 marks. So we're focusing on new cases. So that means 1, we're looking at the bars only. So 2, we're looking at the y-axis on the left. So we need to give two conclusions for this. Well, because there's male and female, that's one way that we can think about it. How are males different to females from this? Of course, we can just look at the trends also and see what happens with there. So let's look at the trends first. So how does the pattern change as the years increase? Well, from 15 to 19 and going on, there's an increase in both males and females up until 65 to 69. After that, there is then a decrease. So that could be one thing that we can say. The next thing that we could do is we could look at the bars and see, comparing males to females, how is it different? Well, for males, they seem to be lower than females from 15 to 19 all the way to 55 to 59. After that, from 60 to 64 onwards, you can see that the number of cases for males is higher than females. The last thing that we could see is, where is it highest? So which year groups have got the highest number of cases recorded? For males, we can see that it's 65 to 69. And it's also the same for females. So both male and females have got the highest number of new cases in that age 65 to 69 bracket. So we have looked at a few points, but remember we only need to state 2. So we can't put more than 2 or else we could lose a mark. So we can say first, from 15 to 59 years, the number of new cases for females is greater than males. And then our second conclusion can be that the highest number of new cases for males and females was from 65 to 69. 5.4. The data for the number of people with skin cancer is given per 100,000 population. Suggest why the data is not given as the total number of people. So it says that each of those points that we can see on our lines is for 100,000 people in the population. So why isn't it for the total number of people? Well, the country that is looked at for this study is in the UK. So the point might be that if we do it for 100,000 people in a population, we were able to compare this data to those in other countries. So we could compare to 100,000 population in Ireland, for example. Figure 10 is repeated below. Okay, exact same graph. So that means that we're going to need to use it again. 5.5. Describe two trends shown in figure 10. Use only the data for the number of people with skin cancer per 100,000 population. So we're doing a similar thing to above, but this time we're focusing on the lines, so on the y-axis on the right. So let's look at the trends then. So starting from the beginning, 15 to 19, what can we see? We can see that the number of females and males are both increasing. And this increase continues up until 55 to 59 years, where the numbers are the same. Then after that, up until 84 years, you can see it's both increasing again. However, for males, the line is steeper. So as we said before when we were looking at lines, because the line is steeper, the increase is faster. So we can say the number of males with skin cancer increases faster than females. So we could say then for that first section, because the whole graph is showing increase, we can say that the increase is quite similar between the males and females from 15 to 59 years. So again, we've got a few points that we could add. So let's start putting our answers down. So our first point then, the number of people with skin cancer between 15 and 59 is higher for females than males. Yes, but it is, remember, it's slightly higher as they increase. And then our second thing we could say is the number of people with skin cancer increases faster for males than females from 59 years to 84 years. 5.6. The estimated population of males aged 80 to 84 years was 694,000. Calculate the number of males aged 80 to 84 years with skin cancer in that year. Use figure 10. Give your answer to 3 significant figures. So they've told us specifically where we need to look on our graph. We know that we're looking at males, we know that we're looking at 80 to 84 years, and we know that we're looking for the number of people with skin cancer per 100,000 population. So again, y-axis on the right, we're looking for the dashed line on our graph. And just a reminder that our total is 694,000. So if we look at that line then to the y-axis, can compare it and then figure out what value that is. So looking at the value, it's going to be 118. 118. So that means that from 118 in out of 100,000 in our population, that's the number of cases that we have. However, we know that the population actually was 694,000. So what we've essentially got to do is we need to think about what's that factor from 100,000 to 694,000. So that would actually be a factor of 6.94. So it's gone up 6.94 times. So what we would just need to do to find out the number of males in a population of 694,000 is take our value 118 and multiply that by 6.94. And doing that, we come up with a value of 818.92, but the question is asking us to give our answer to 3 significant figures, so we know rounding that it's going to be 819. 890 number of males with skin cancer. Question 6. This question is about the heart. 6.1. Why is the heart described as an organ? 1 mark. Well, it's just asking us then to define organ. It's made up of different tissues, just like a tissue is made up of cells. 6.2. Valves in the heart keep the blood flowing through the heart in one direction. Figure 11 shows the heart with one of the valves labeled. Explain the effects on a person if the valve labeled in figure 11 developed a leak. So a valve, what's the job of a valve? Is to prevent backflow. So it causes the blood to just flow in one direction only. So if the valve developed a leak, that would then just mean that the blood would flow backwards. But let's remind ourselves the valve is labeled specifically in that part of the heart. So we know that we're dealing with the left side of the heart. And thinking about the passage of blood, we know that the blood is going to go into the heart for the left side. So that's going through the pulmonary vein. Then it's going into the left atrium, down into the left ventricle, and then through the aorta out of the heart. So if this valve is leaking, that means that some blood would fall back into the ventricle. So it wouldn't be able to leave through the aorta. Now remember that the question is asking us to explain the effects on a person. So what would be the consequence of blood not leaving the heart? Well, if it's the left side of the heart, we've got to remember that that blood is going to be oxygenated. So it's just come from the lungs and wants to go to the rest of the body. So if this blood is falling back, that means that less oxygenated blood is going to the rest of the body. And then that means that less oxygen will reach the tissues, because all over the body are tissues, where the muscle and some of the other organs, for example, and they all need oxygen. Why do these cells need the oxygen? Well, if we talk about muscle specifically, it's going to need the oxygen for it to contract. But how does it contract? It's through respiration. And if it's dealing with oxygen, it's going to be aerobic respiration. So if less oxygen is reaching the tissues, there'll be less aerobic respiration carried out. But the cells still need to respire. So that means that instead of aerobic respiration, it'll be anaerobic respiration which will be performed. And thinking about bioenergetics, what's the effect of more anaerobic respiration occurring? You're going to get a buildup of lactic acid, which leads to muscle fatigue or muscle cramps. So this is a very lovely question that the examiners love to put in, where you're able to link your knowledge of organization, structure of the heart, to bioenergetics and the idea of respiration and lactic acid. 6.3. Faulty heart valves can be replaced using biological or mechanical valves. The faulty valve is replaced during an operation. Biological valves are from animals or human donors, allow blood to flow through them normally, wear out and stiffen over time, so they may need to be replaced. Mechanical valves are made from synthetic materials, may cause blood clots on the surface of the valve, require anti-clotting drugs to be taken for the rest of the patient's life, can last for a very long time in ideal conditions. A young woman enjoys extreme sports and would like to start a family. The woman needs a heart valve replacing. describe the advantages and disadvantages for this young woman of having a biological heart valve instead of a mechanical heart valve. Right, so we're thinking about the pros and cons of biological heart valve instead of mechanical heart valve. So straight away we can separate our space now into the advantages or plus points for biological valve and the negative disadvantages of the biological valve. So we need to just focus our answers on those two areas specifically in order to try and get 4 marks. So we need to go back to our information about the two valves, and then we need to figure out what are the plus points and what are the negative points about them. Remember that we can't just take the information from here and put them down in our answer. We need to go in a little bit more in depth about what they might be linked to. So the first thing that we can say from here, the biological valves are from animal and human donors. So what's the benefit or point about that? Well, if they're from an animal human donor, there is a little chance of rejection. And the reason is because the antigens that are present on these valves, most of the time they are removed, so there's less chance of it being rejected by the immune system. Next point, it says wear out and stiffen over time, so may need to be replaced. Well, if they need to be replaced, then that would mean that you may need to wait for available donors because they're not going to be there all the time. So that can be an inconvenience and it can prevent you from doing certain activities. So already the little chance of rejection, that can be at a positive point for biological valves. May need to wait for available donors is definitely a negative point. So next thing that we have to think about is the mother would like to start a family. So we need to think about how that might be linked to any of the points that we've gotten so far, or any points that we're about to get. So the second point from our biological valves, it says allow blood to flow through them normally. Now, during an operation, surgery, there is always a chance of blood clots forming. So, because these biological valves are able to allow blood to flow normally, that should mean that that should reduce the chances of blood clots appearing. Remember, and because this woman would like to start a family, she might have to go through pregnancy and birth. So, having blood clots could be a problem for not only her, but for her baby. So, by allowing the blood to flow through them normally, that means that there should be a small chance of this happening. Okay, so we've analyzed everything from our biological valve. Let's look at our mechanical valve now. Are made from synthetic materials. Well, there won't be any antigens on a synthetic material, so that means that the chance of rejection is going to be 0. It says may cause blood clots on the surface of the valve. So as we said, could lead to blood clots during the pregnancy, could lead to blood clots during the birth, which is a very serious problem. Third point, require anti-clotting drugs to be taken for the rest of the patient's life. So the mechanical valves, as it said, cause blood clots. So to try and reduce that from happening, you need to take these drugs to cause that to be reduced. But there is a potential that side effects may occur during the taking of this drug, which could actually affect the baby if she's pregnant. And the problem is, as it says, she enjoys extreme sports. So if she needs to take these for the rest of her life, that would mean whenever she's going on these adventures for extreme sports, she would need to carry them all the time, which again is a little inconvenient. And then the last point, can last for a very long time in ideal conditions. Well, that should be a positive point. because it means that they would be less likely to be replaced. So we've analyzed the points now, but we need to remember that we're describing the advantages and disadvantages. Now, this is sort of an evaluate question, but a little bit different. We know that we need to provide the pros and cons for each, but what we still need to do is we need to compare, because when you find advantage, it is over something. So it must be some comparative language used. So let's look at our points one more time, and then let's see if we can change some of our answers or change some of our wording so it is more comparative. So if we look at the first points for biological mechanical, it's to do with rejection. So our mechanical valve, there's no chance of rejection, but for our animal, for our biological valve, we said there's a little chance of rejection. So what we can do then is we can just change the word in. So it says there's a higher chance of rejection with our biological valve, which means it's not actually positive anymore. It's actually a negative point comparing it to mechanical. We know that we still have a negative point with this needing to wait for available donors. But then we were saying that the point is for allow blood to flow through them. Normally, we wrote down that it was a little chance of blood clots during pregnancy. Well, what we can do then is we can change that to a lower chance of blood clots during pregnancy compared to mechanical valve. So that actually is then a positive point. So if we look at our biological valve only, then we've gotten one positive point and two negative points. So we have found advantage and disadvantage. So when we were thinking about this higher chance of rejection, what that probably means then is that we would need to try and somehow reduce the chance of this happening. So what that would then mean that the person would need to do is that they would need to take drugs to suppress or lower the immune system's response, which again is a negative thing. So let's list our points now in the relevant places, see how many we've got on both sides. So if there is a very little chance of blood clots forming, then that would mean then that we wouldn't need to take any anti-clotting drugs, which is definitely an advantage. Another advantage then is there's a lower chance of blood clots during pregnancy or birth. So we definitely got at least two positive points for our biological valve. Let's list our negative and disadvantages for our biological valve now. So first disadvantage, it may be rejected, so you may have to take drugs to suppress the immune system. The drugs that have to be taken may have some side effects, which could affect the baby, could affect the mother. You may have to wait for a suitable donor because they're not always going to be available. And the last point that it was talking about may need replacement. So that would mean that you'd have to go through multiple operations, multiple surgeries to replace the faulty valve when it's time. And of course, we did say that any time that there is some sort of operation, there is an increased chance of blood clots forming.